I am going to be switching gears. We're going to be focused on the gut now, right before lunch. I think you all have given my background slide, so I'm going to go pretty quickly through this. We all know why we're here. I think this convergence of age and HIV-related comorbidities is really critical. Our lab is interested in understanding mechanism so we can develop targeted therapies. This is kind of an old slide, but it's to remind me that to stress how important this is clinically, as we've heard from a number of you. I guess it's estimated that around 70% of persons living with HIV infection may be over 50 by 2020. And we've heard from a number of other people here at the meeting that many of these individuals will have multiple comorbidities as well. So our group's been interested in inflammation and what drives inflammation. And it's important to note that there are a lot of commonalities among the type of inflammation or markers of inflammation in HIV and in those independently reported in aging, which is often called inflammaging. We've gone through these on a number of other occasions, but there are increased cytokines in the plasma, acute phase reactants, procoagulant factors, markers of both monocyte and T-cell activation, and of course there are a number of shared clinical outcomes that have been linked to inflammation independently in both of these settings. So again, we're looking at whether there might be common mechanisms that drive inflammation in HIV infection and aging. And I'd like to try to convince you that the gut is an important source of inflammation in both of these clinical settings. So in terms of the gut being disrupted during HIV infection, I think this is pretty well understood at the molecular and cellular level about what's happening. And HIV replicates in the gut tissue at very high levels during the earliest stages of infection, targets receptive CD4 T cells, kills them, creates a lot of inflammation. This ultimately disrupts the epithelial barrier, but there is this associated microbial dysbiosis, or change in the gut microbiome that's been linked to all this gut pathology. And one of the major consequences of this mucosal pathology is microbial translocation. And we've heard a lot about that, linking that to a bunch of other comorbid conditions. But early on, it was documented that bacterial LPS was elevated. This was linked to T-cell activation and disease progression in untreated HIV infection. More recent studies have shown that microbial translocation persists despite effective suppressive antiretroviral therapy. And its persistence really predicts both morbidity and mortality. So I think a little bit less is known in the aging field about the gut, but I'd like to provide some data to support that the gut is also disrupted, or homeostasis in the gut is disrupted during the physiologic aging process. So we published in 2014 a paper looking across the age span from like age 20 to 100 years of age, and these were uninfected participants in a study. They were our control participants. And we looked at inflammatory biomarkers in their association with age. And we found that gut-related biomarkers, so intestinal fatty acid binding protein and bacterial LPS, a marker of microbial translocation, also increased with advancing age, as did these other markers of T-cell activation, monocyte activation, and systemic inflammation. And importantly, these markers, these gut-focused markers, were directly correlated with the cellular activation markers. Again, suggesting there's something going on as we age that's causing a leaky gut. What we don't know is what's causing this, and there's a lot of interest, I think, in the aging field about this. And some of the possibilities range from ischemic stress from hypoperfusion, hardening of the arteries and low blood flow, to epigenetic cellular changes in homeostatic signals, toll-like receptor, Ni-like receptor expression that are involved in homeostasis. There is a possibility that there's lamina propria inflammation or immune cell dysfunction, so immune senescence might be contributing at the gut level. Loss of mucin and antimicrobial peptides have been postulated as well. But there's growing evidence that the changes in the enteric microbiome, or dysbiosis, as we call it, is playing a role also in the age-related inflammation. And this is a review, if you're interested in this topic, it's a really good review on age-related inflammation in the microbiome by Thomas Buford in 2017. And what he found, let me see if I can get my, look at that. What he found was that aging and inflammation and the microbiome have independently been linked to a number of comorbid conditions that look awfully similar. And so he said, it's not really a stretch to think that these things might be in a common pathway. So I want to kind of switch gears now and really focus in on the microbiome. For those of you not familiar with the concept of how we measure this and what this really means, I'll just do a brief review. We're really talking about communities of bacteria that kind of live in harmony. Some people call it a separate organ in our bodies because there are 100 trillion microbial cells. And they're unique to each individual, but if you look between individuals, the taxa themselves are very different, but the functions are pretty much the same. So they have a lot of different jobs that they do. So they're involved in nutrient metabolism, maintaining that barrier function, which is really critical, and then in all aspects of immune function, from development of the immune system to the mucosal immune system to more systemic processes. And importantly, and as a confounder for those of us studying the microbiome, there are a number of factors that influence it. And so diet is critical. Sexual practices are important, and there are numerous other factors. Drugs, or I should say therapeutics, especially antibiotics, obviously play a role. Exercise may be a factor as well. So it's hard to control for all these kind of confounders. I want to talk a little bit about not only the bugs themselves, and I'm going to focus on bacteria today, but their metabolic products, and they're incredibly important and bioactive. And short-chain fatty acids are something that we've been involved in studying, and I'll be talking more about those a little bit later, so I want to go back through a background on that. So short-chain fatty acids are metabolic breakdown products of dietary fiber, and they're fermented primarily in the colon by anaerobic bacteria. You've probably heard of propionate, butyrate, and acetate. They're the three major short-chain fatty acids, and they play really important roles both at the gut level and more systemically in lipid and glucose metabolism. The primary role of butyrate is actually to provide energy for enterocytes. But butyrate has also been shown to have immune modulatory or anti-inflammatory properties, and that's kind of where our interest lies. But importantly, butyrate is produced primarily by firmicutes. It's a specific phylum of gram-positive bacteria, and we'll talk more about that in a minute. So a lot of studies have been done independently looking at the microbiome and dysbiosis and HIV infection and in aging. And I don't have time to go over all the specifics of those studies, but what I'd like to do is kind of summarize some general trends and commonalities among them. So in general, there tends to be an increase in gram-negative bacteria that have pathogenic properties. And many of you will be familiar with some of these. They tend to be, we call them pathobionts. Enterobacteriaceae, things like E. coli and Enterobacter, for instance, if there are any ID physicians here. And then a decrease in bacteria concurrently that have anti-inflammatory properties or considered sort of healthy bacteria. These tend to be gram-positive bacteria that either make short-chain fatty acids or you might know as probiotics, so bifidobacteria, lactobacillus, and the like. In addition to changes in the taxa abundance, there are also changes in the diversity of the species, and typically you get a decrease in the diversity with each of these conditions. Now, importantly, again, there's starting to be studies that link dysbiosis to a number of different comorbid conditions and inflammatory readouts. And again, I just want to kind of briefly go over these. Dysbiosis with age has been shown to be linked to frailty indices, to inflammation levels, to neurocognitive dysfunction. And importantly, these again are usually with positive associations with pathobiont bacteria and negative associations with or loss of short-chain fatty acid production. Similarly, in HIV infection, the microbiome or changes in the microbiome has been linked to disease progression and immune status, as well as all of these readouts we talked about. Microbial translocation, barrier breakdown, systemic inflammation. And likewise, with some differences, there have been changes, increases in these proteobacteria or pathobionts, decreases in short-chain fatty acid producing bacteria. I don't want to get into, there's a bit of a controversy about the role of Prevotella that has been linked to sexual practices and is kind of confounding the field right now. But it has also been associated, increases in that particular bacteria have been associated with inflammation. So we identified as a big knowledge gap the fact that really very few, if any studies, I don't know of any studies that have really looked at the microbiome and changes in older people living with HIV infection. And in particular, whether dysbiosis, that's age-associated dysbiosis, is actually different if you have HIV infection or not. And so we set about to study this in a collaborative effort with Christine, my partner in crime up here, who was actually enrolling patients into her exercise and healthy aging study, as well as Christine Seifert, who was doing a pharmacology and aging study, both at University of Colorado. And so we enrolled into sort of a sub-study, a very pilot study, some of those individuals who were aged 50 to 75, and the HIV-infected cohort were all suppressed on antiretroviral therapy for at least 2 years, and we had a seronegative control cohort as well. And we obtained stool samples and dietary information from those individuals and did a microbiome analysis with standard 16S ribosomal RNA sequencing. We also looked at stool short-chain fatty acid levels for the reasons that I previously discussed, and systemic biomarkers. And these data have not been presented before, so again, be kind, as our former colleague mentioned, but I'd love to get feedback from you all. And what I'd like to do is present some of the data, not focusing on the specifics of which bacteria went up and down, because we could spend all day on that, but really on some of the big picture kind of concepts that I think that come out of this data set. So the study participants, there were 14 people living with HIV, 22 controls, so a pretty small sample size. They were well matched in terms of gender, age, averaging around 60, mostly Caucasian, and body mass indices were similar. Where we weren't able to match well had to do with sexual practices. And that might become an issue in terms of interpretation of the data. Again, everyone was, the HIV-infected cohort was suppressed. CD4 count was close to 600 on average. Just very briefly, we did not see differences in alpha diversity, what we call alpha diversity, which is basically kind of counting up the number of taxa that you see between our infected and uninfected cohort. However, when we looked at beta diversity, and that is really looking at the abundance of different bacterial taxa, we did see differences. So if you just look at a straight analysis of infected versus uninfected, you start to see differences. And as you go down the taxonomic rank from phylum to family to genus, you start to see more differences. As a whole, they weren't statistically significant. And our statisticians really felt that we were not, with the sample size this small, it was difficult to really get at the importance of the HIV effect using this type of analysis. So they went further and used generalized linear models to address the effect of HIV within this cohort. And we did indeed find significant HIV-related differences in the microbiome at every level except for the phylum level. And you know what I'll do here is just point out some findings. So here is Enterobacter. If I can get this, that's right there. Enterobacter went up as a result of HIV infection, and then you can see going down, sorry. Roseburia down here is a short-chain fatty acid producing bacteria that was reduced. Many of the findings here mirror what has been done with in other studies, so nothing really surprising, but the important point I think is that in this aging cohort, we do see HIV-related effects. Now, it's important for us to look at the effect of age, but you might say, well, you don't have a young group, so that's a problem. But our statisticians were able to look at the effect of age across the age of 50 to 75, and this analysis looks at the impact of each year of increasing age on the microbiome. And we did find significant age-related effects in these cohorts. They were a little bit counterintuitive, and what we found in general was that some of the markers or bacteria that I typically were increased with age when you compare young to old cohorts were going in the opposite direction in this kind of study. And I can talk to you about why I think that might be offline, but it was a little bit counterintuitive. So for instance, we saw increases in lactobacillus, which we think of as a healthy bacteria with increasing age. We saw decreases in Enterobacter, which I said is a pathobiont with increasing age. So those things are a little bit counterintuitive. But we did see age effects. Now, we wanted to then look to see whether there were actually interactions between HIV and age. And the way this analysis was done, it was to look at whether HIV serostatus impacted the age-related effects on the microbiome. And so here what I'm showing you is just, this is the age effect among people living with HIV infection. And the red with the increased arrows means that these bacterial taxa were increased with age among people living with HIV infection. You can see that some of these are pathobionts, like fusobacteria, for instance, which again tends to cause infections in people. However, when you look at the age effects in our control group, you can see that there are very different patterns. And there are positive interactions, age and HIV interactions. In the take-home message here, we're still parsing through, what it means for this one to go up and this one to go down. But the bottom line is that the geriome, or the age-related microbiome, is different in older persons with HIV infection. So we can't always assume that what we see in older individuals or therapeutics for older individuals will necessarily be effective or will be the same in people living with HIV infection. We next looked at systemic biomarkers, and in this cohort, we did see increases in HIV-related increases in soluble CD163, soluble CD14, those monocyte activation markers, and the soluble TNF receptors. We saw increases in other markers. IFAB was elevated, but didn't reach statistical significance. And what I'm showing you here is our attempt to start to look at associations between inflammation markers and the microbiome. So we took on the right hand side here, these are all the genera, or the bacterial top 25 genera. And then if you look along the bottom, you can see we have all the inflammatory markers. And this is a heat map. So red means positive correlation, blue means negative correlation. The stars indicate, or asterisks indicate significance. And so what you see, if you just look at this and eyeball it, you can see they're very different patterns, okay? And I've put boxes around some of the areas where you start to see significant positive associations. And you can see that if you go over from the controls, for instance, and you look at the positive associations here, and you look at the HIV-infected group, you see there are actually, many of them are actually going in the opposite direction. So again, we're trying to sort out what all these associations mean, but it's important to know that not only is your microbiome with age different in HIV infection, but your associations or your relationships between the microbiome and inflammatory markers differ as well. We looked at stool short-chain fatty acids, and our hypothesis is that these would be lower in persons living with HIV infection, because we've previously shown that some of these short-chain fatty acid-producing bacteria are decreased in HIV infection, and we found similar results here in this cohort. However, that's not necessarily what we found. So except for propionate, which trended lower, we didn't see differences in short-chain fatty acids between our two cohorts. Butyrate actually was higher in the HIV-infected cohort. But when we looked at dietary intake, it's possible that some of that can be explained by the fact that our HIV-infected cohort tended to eat more fiber than the negative cohort, and so there's a balancing effect there. So it's really important to look at diet in these microbiome studies, because I think a lot of the findings might relate to dietary habits. So again, lastly, we looked at correlations between the short-chain fatty acids in the stool and dietary features and the microbiome. And again, just eyeballing this from where you're sitting, you can see that there are very different patterns in the uninfected group, control group, and in people living with HIV infection. Again, suggesting that relationships between what you're eating, between the amount of short-chain fatty acids in your stool and the microbiome differ depending on whether you have HIV infection. So I just want to conclude by saying that we did find both age and HIV effects independently in this older cohort of individuals, and that the relationships between the microbial dysbiosis that we see in older individuals and a lot of these other factors such as inflammation, diet, short-chain fatty acid levels differ depending on whether you're HIV infected or not. And of course, the study limitations, including small sample size, a limited age range of 50 to 75 without a young control group, or younger control group. And then we also didn't specifically collect data on probiotic use. Christine did a very nice medication review, but not everyone considers probiotics a medication, so that it wasn't reported very often. Now, I just want to very briefly mention, in terms of future directions, and try to wrap this back around to frailty and sarcopenia, perhaps, is that we're planning to analyze and look for associations between the microbiome and microbial, some of these metabolites that we talked about, and features of physical function that Christine and her group collected. And so I included a little bit of a teaser slide here. So what you see in the slide is that higher levels of stool butyrate correlate with greater grip strength, okay? So if you look at butyrate on the y-axis, grip strength on the x-axis, you can see there's a positive correlation among the entire group. Now, we're not sure exactly what that means. However, it could mean that eating more fiber makes you stronger. I don't know. But we're going to begin to look at some of these associations and hopefully, ultimately, start to look at the gut-muscle axis and try to understand what it is that's mediating some of these links that have previously been seen with frailty. We also are evaluating currently the effect of exercise on the microbiome and in older people living with HIV infection. So we hope to have more information for you on that very soon. And then lastly, my lab is interested in looking really at the cellular level, at the mucosa, to understand the interactions between the microbiome and aging cells, and how this relates to inflammation. So hopefully future studies will be coming on that. So I want to end by acknowledging and thanking all the members who have worked on this in my lab and Christine's lab, especially Jay Liu, who was a fellow that Christine and I co-mentored, Steph Dillon, who's worked very hard on this as well. Dan Frank, Miranda Kroll, and Rachel Johnson have been critical in terms of the microbiome analysis. Janine Higgins did our dietary analysis. And then from Purdue University, our collaborators, Bruce Hamaker and Eunice Tunzel, measured short-chain fatty acids for us. And then I want to thank the financial sponsors of our study as well. Thank you.